terms of an introduction to drug discovery, what I'll be doing is just giving a little bit of a flavor of what you can expect for the rest of the week. So going through the drug discovery pipeline, which some of you may be familiar with, but also introducing some of the AI concepts into that pipeline, and then diving deeper into what type of screening methods are available, just a brief introduction into ACME and DMPK, where Matthew and Lisa will go more in depth on Thursday, and then touching a bit on chemical properties. And then I'll hand over to Jason, who will go into the examples of how we've used AI and machine learning at H3D. So this gives you a broad overview of the whole drug discovery process. So right in the beginning, you have your basic research, your basic science in pre-discovery phase, and this is usually done in your research institutions. And then taking it through to the drug discovery process is quite extensive. So in order to get one approved drug at the end, you go through lots and lots of compounds, and as you go into preclinical trials, that amount of compounds get reduced, and then furthermore, that attrition goes off and off and off until you land up with your one compound that's going to make it at the end. And you can see it takes really long within each of these phases, and uses a lot of volunteers in the different phase of clinical trials, and all of this increases the costs. If you have more compounds, if you have more people, if you use more animals, your costs are just going to increase and the time increases. And the average cost, there was a study that was done that's indicated at the bottom of here, that the average cost to get one drug to market is around $1.3 billion. So it's really an expensive process in order to find that one at the end. And so how do we go about doing just this drug discovery side of things? So I'm going to zoom into just this phase, and that's what we're going to be speaking a bit more about this week. So over here, I've got our generic pipeline, drug discovery pipeline, which you may or may not be familiar with. So I'm going to take you through stepwise how you get all the way to your clinical development. Where you're going to start is with some screening. So in screening, you're generally using a compound library, and this is usually quite high throughput, usually at single point assays. And then you want to prioritize which active hits you're going to go with. So you will do a SWOT analysis. And then once you have your screening active, this may come from different plates. It could be in a solution that's been in storage. So you want to retest a fresh sample to be able to validate that hit. And this is where you'll most likely do dose response assays, and also get an idea of what the ACME properties are going to be. So this is looking at your solubility and your toxicity, and maybe look at a bit more of the biology behind it so that you can put your resources into that hit. Then once you have your hit, you want to scope around the molecule. Can you chew off parts of the molecule, make it a bit smaller? Can you add bits? Can you move bits around to be able to understand what the different structure-activity relationships are? And to be able to really define that minimal pharmacophore, the minimal structure that will get your optimal activity. So this is about 30 compounds. Once you have your hit series, then it's exciting times where you get into hit to lead and lead up. And in hit to lead is where you're really trying to improve that potency, as well as improve some of your other properties of your molecule. And in this, you want to be able to get your proof of concept. So this is where you've tested in vitro, you've tested it in all your assays. Does it translate into an animal model? Can you get an idea of how it's going to behave in the body by doing pharmacokinetics? And in this phase, you kind of want to get a few compounds that tick some of the boxes, but you don't need to have all the boxes ticked in one molecule just yet, because that's what lead optimization is about. So here you're really trying to optimize for that potency, optimize for your properties in order to get your best front runner molecule, usually about one to three front runners that you can then put forward into candidate profiling. So within lead up, there's a huge focus around toxicity studies, as that's a cause of attrition. You also want to understand how your PK corresponds to how you get your effects in your animal model. And you're also scaling up. So you may start your PK in a mouse, and then you move to a rat, a dog, or a monkey. And this is in order to be able to predict how it's going to behave in your human species. When you get to your candidate profiling here, you've got to scale up your compounds a lot because you're doing lots of advanced studies, and this is often done in your different species, as well as refining those human dose predictions a bit more so that when you go into clinical development, you have an idea of what dose you're going to give. So this gives you an indication of the general generic drug discovery pipeline and the types of activities that are involved in each of those. And now to give you a flavor of how AI fits in within this pipeline and where you can start inputting that in order to increase the time, decrease your time and decrease your costs. You can do these types of activities at these phases. So within the early stages, you can use virtual screening. So if you know your target, you can run using computational methods. You can run lots of different compounds through it to choose only the best compounds that you want to take forward into synthesis and retesting. You can also use computational tools to do scoring profiles to be able to triage and choose which series to take forward. And then this is also a good time to evaluate your chemical space. Where within the lots and lots of compounds out there does your series fit in, and is that a worthwhile space to pursue? And Jason will dive deeper into that today. Then later on, in hit lead and lead up, you do quantitative structure activity relationship models to be able to predict your biological activity. And this is what Gemma and Miguel will go deeper into, is how do you build models to be able to predict how active and potent your compound is going to be. There's also various tools to be able to calculate and predict what the admin properties are going to be, what your solubility will be like, or metabolism. So really using computational methods to predict and calculate to avoid too many compounds being made unnecessarily. The space is also developed into being able to actually design compounds for you, which we'll touch on a bit later. And then also there's a lot of tools to be able to predict physicochemical properties, which I'll touch on later in the presentation. What we won't go into too much is around the clinical development side, and this is where they really use AI machine learning tools to help the clinical trials in designing, in monitoring patients, and in recruiting patients as well. So there's huge scope for using these methods to be able to expedite the processes. So all of this AI machine learning, how does this help? So this helps in reducing that initial cost. It helps to speed up our timelines, and hopefully also reduces the number of compounds that we synthesize, so that we're really prioritizing what compounds get made and putting our best foot forward. And ultimately also hopefully reducing the number of animals that gets used in later studies. So focusing in on the early stages when you are doing your screening, some of you may already be doing some of these types of biological assays in your research institutions. So one method is called phenotypic screening. So this is when it's a whole cell assay and you assume it's going to be relevant in the human later down the line. Because it's the whole cell, you don't know what the target is going to be. So later on, you've got to deconvolute what that target will be. And this is just an example of a dose response plate for TB. In a target-based approach, this is when you know what the target is that you want to aim for. And then you can screen different libraries against it, and you can see which one fits in the best way. So then you'll need to develop a biochemical or enzymatic assay to be able to screen your compounds against. But this is where you assume that the mechanism of action is actually going to translate into the whole cell. Another method is fragment-based. So this is where you have the compounds are a lot smaller, they're small little fragments, but they bind a lot better in those pockets. There's a better fit. And then later on, you can link those different pieces together to form your one molecule. And so even though you may get weak potency for these individual little fragments, when you put it together, it should be high binding and get good potency. Another screening method is using natural products. Definitely on this continent, it's something that we explore a lot. And this can either be from plants or microbials. However, isolating the actual active ingredient can be quite challenging, but one can do semi-synthesis on that to be able to optimize the properties of that natural product a bit better. But the method that we are applying in this context to help speed up all of these is virtual screening. So virtual screening is when you have a known target with your crystal structure, and you can dock your molecules in that, and then it goes through, you can apply different filters through to try and prioritize which and select which compounds you're going to make. So you can see over here, they have a few molecules, but at the end there's only 3 where they'll synthesize that and then test it in an in vitro setting to be able to see whether what they have docked early on actually is proven in the in vitro assay. So switching over onto ADME and DMPK, I'm just going to briefly go through this. I've said ADME, the acronym, a few times, so for those of you who don't know, the A in that is related to absorption. So how does the molecule, you take it orally, how is it going to get into the bloodstream? How is it going to be absorbed? The D is distribution. How does it get to the site of action? How does it get to where it needs to go around the body? The M is metabolism. Within your liver, you have enzymes that break down molecules. How much metabolism is happening to that compound? And with that metabolism, it then gets excreted out your body because you don't want to build up of toxins in your system. So we have a number of assays that help to measure these, and these are a group of in vitro assays, so things that you can do in the lab, in a test tube. First one being solubility. How soluble is the molecule? Is it going to be able to get into the water-based systems in our body? Second is permeability. Will it be able to permeate the membranes in our body? In our body, we've got all sorts of different organs and things that it needs to get through, and how's it going to eventually get into that site of action? We also have lots of protein in our body, in our blood, and so only the amount of compound that is available that is not bound is what can actually cause an effect. And so we have a method of measuring the plasma protein binding, so how much free drug is available for action. Then in terms of measuring metabolism, there's an assay for metabolic stability. How stable is your compound after 30 minutes after being exposed to these enzymes? And we can do it in different species, mouse, rats, and humans, and that gives an indication of how it metabolizes in the different species. Sometimes your molecule may get chewed up very, very rigorously, so you want to identify what is the metabolite, how can we stop this in future molecules that we make, so that you can do MET ID. And then there's also these enzymes are called CYP450 enzymes that metabolize your molecules. And sometimes compounds can inhibit these CYP enzymes, which is a problem if you have another drug that gets metabolized by that. So then you can get drug-to-drug interactions. So if the other drug that is being taken doesn't get metabolized, then it can build up in the body and cause a toxic effect, which you don't want. And then more generally, looking at cytotoxicity, how toxic is it to the human? Is there selectivity for your parasite or your bacteria over the human cells? So that's what cytotoxicity measures. And then quite a big one that is a cause for attrition is looking at cardiotoxicity, and that's measured by this hERG assay, which stands for human ether egogo related gene. And this is just an indication of whether the drug is likely to cause heart arrhythmias. So you don't want that in a human or an animal, so you're able to measure this early on in an in vitro setting. So now I'm moving over from an in vitro setting into in vivo. That's what's called pharmacokinetics. And this gives you an indication of how the molecule is going to be distributed in the body. So over here, I've got a very normalized illustration of a PK curve. So here you've got the concentration of your compound on the y-axis, on the x-axis is time. So usually you'll do both an oral administration as well as an intravenous administration. So if you're giving it orally, the drug has to get past all the membranes and be absorbed to eventually get into the blood. Whereas with intravenous, it gets straight into the blood. So that's why you get the difference in the curves. And then using this difference, you're able to get various variables, which Lisa and Matthew will dive deeper into. So this illustration indicates over here is your in vitro assays that I mentioned. You've got your solubility, your permeability, metabolic stability, and your binding. And this all relates into what you're going to get in your in vivo setting. So permeability and solubility is a measure of your fraction absorbed. Metabolism is related to clearance, and binding is related to volume of distribution. And ultimately, the values that you get out from a PK study is your bioavailability. That gives an indication of how much of the compound, how big is the dose that you need to give. And then your half-life is when it's getting eliminated, how frequently do you need to give that dosage to the human. So that's all I'm going to touch on PK and ADME, because we'll hear more about it, but hopefully that's giving you some of the context that we'll get into. And there's certain properties of a molecule that affect these ADME and DMPK properties. And if you're not a medicinal chemist, that's okay. You don't really need to know too much of what these chemical properties mean, but you can know that there are ways to predict them, and there's ways to understand the trends so that you can be able to apply those learnings going forward. So I'll do my best to try and just briefly touch on what these chemical properties are and how they affect the assays. So polarity is how polar a molecule, compound is, and usually this is the polar atoms are oxygen, nitrogen, phosphorus. So if you see that there's lots of those in your molecule, you can get an indication that it's probably quite polar. And we usually use topological polar surface area, or TPSA, as a way to measure that. And usually increasing polarity results in an increase in solubility, which is a good thing. But it also results in a decrease in permeability, but it also helps with improving your hERG. So you'll see I've indicated arrows which show whether the value will increase or decrease, but sometimes that means it's a good thing, sometimes that means it's a bad thing. So I've color-coded it to make it a little bit easier. So with lipophilicity, this is you can either measure lipophilicity or calculate it using log D or log P parameters. And this gives an indication of whether it's able to mix with fats or not. And this is quite a big driver for a lot of properties that you get out. So it's usually quite a good one to use to see whether there's any trends that you can pick up and try and modulate, because it's quite an easy one to to do structural modifications with. So increasing your lipophilicity, it's gonna result in poor solubility, but it does help with your permeability. It's likely to increase your metabolism, but can be detrimental to your HERG. But you generally do get an increase in potency, but it's kind of a superficial increase. So you can see how you kind of need to balance all these properties in order to get the best mixture of all your in vitro assay results. Another one is basicity, and usually you can use pKa as a measure of the strength of the acid, and generally adding an ionizable group like a pyridine or nitrogen. This results in improvement in solubility and permeability, but is usually not very good for HERG. The next one is hydrogen bonding. So you can see over here, this is an amide, so there's a nitrogen here with hydrogens. So usually the hydrogens can be a hydrogen bond donor, and then these oxygen has these two little lone pairs, and then it's a hydrogen bond acceptor. What you need to know from this is this causes binding to your active site. So there's lots of other hydrogen bond donors and acceptors in your enzyme that these molecules can bind to. Therefore, having more of that generally results in an increase in your potency if it's in the right place, but also increases your HERG, which is a good thing, decreases permeability, but increases your solubility. And then for molecular weight, this is your size of your molecule. It's really usually quite easy to determine. And when one starts on a project, it's generally quite small, and then you build on. And so increasing that molecular weight can sometimes result in detrimental to solubility and permeability. But once again, you tend to get more binding because it's bigger, pulls up the space more, so you get an increase in potency, but sometimes also superficially. So there's this, you may have heard rule of 5 or rule of 3. It's more of a guideline, but this is general principle for drug-like properties. And this looked at drugs that are already on the market. So your ultimate compound at the end of the day, you want it to have around less than 5 hydrogen bond donors, your polar molecules less than 10, molecular weight less than 500, and your lipophilicity log P less than 5. But that's right at the end. So if you're wanting to start, you probably want to go with the rule of 3 so that you can build up. You can increase your lipophilicity, you can increase those different atoms, and you can increase the molecular weight and be able to get to that sweet spot. So I hope this helps to paint a picture of the different chemical properties that come into play and how it affects your different assays. There's different tools to be able to calculate these and to be able to create visualizations. This is a free open access tool with Data Warrior, and this is what the interface looks like, and they calculate these properties for you, and then you can do different plots of how increasing the lipophilicity may affect your potency or your solubility. And then there are a number of other tools that are available, but however, these are paid for tools. CDD Vault does have a free visualization tool as well, but some of these you can also access through your institutions. So this is a graph of one of those visualizations where increasing your TPSA improves your HERG. And so you can see you get a nice correlation. So you really, with these properties and your ADME properties, you really want to try and understand how your molecules are behaving so you can move towards improving them. So that's the end of all the content from my side, but just to kind of summarize in more general terms. So when you are starting out, you wanting to look at your activity, and you're also wanting to look at your properties. And so you generally synthesize these or get molecules, and then you evaluate it in your in vitro biology assays, as well as your ADME assays and your physical chemical properties. And once you have good in vitro data, then you can move to in vivo. So getting your in vivo proof of concept, as well as your in vivo pharmacokinetics. But you constantly, it's a reiterative process, and you're feeding these properties back into redesigned molecules. And where AI and machine learning can help is in many parts of these, in terms of predicting biological properties or predicting your admin properties, as well as visualizations to be able to redesign your molecules. So I hope that's giving you a little taste of what's to come. There is a book called Drug-like Properties that goes in depth with all these concepts. So it's a really useful manual and handbook to go back to if you're unsure of anything and want to design new compounds or understand these assays further. So that's it from my side.